Okay, students, this is the chapter of microorganisms use in biotechnology. In our previous lectures, we saw the general structure of fungus, virus, and bacteria. We saw their application in biotechnology. We saw how we can make yogurt, cheese, alcohol, and bread from them. And today's we have to see some other applications of microorganisms in biotechnology. The applications, as I told you before, that they are numerous, but here we will focus only on these three ones. This is the penicillin, which is an antibiotic. We'll see how the insulin can be formed using microorganism, and then we will see the formation of SCP, that is single cell protein. All these products are industrially made on large scale, and for that obviously you need to have a bigger container. That bigger container is actually called fermenter. The industrial fermenter which is used has a general features as seen in the slide here. We see that it has a electrical appliance, that is the motor, which is connected with the impeller or stirrer. This motor is outside the fermenter, which has a lot of benefit for us because since motor is an electrical appliance, it is protected from the contents of the fermenter. Plus, since it is outside, it is not affecting the capacity or space inside the fermenter. The maintenance of motor is quite easy if it is outside. And the very important point is that this motor generates heat, which may have increased the temperature of the contents of the fermenter. So we don't want that. We don't want to have greater or higher temperatures, since it may damage our product and also the microorganism that you are using for the formation of your desired product. This impeller or stirrer is used for mixing of the contents of the fermenter. Then we have a pH controller. As the name is indicating, it will control the pH. The exhaust is there to take out all the waste gases, which mainly includes carbon dioxide. Then we have temperature and pH probe, which detects the changes in temperature and pH respectively. This pH and temperature must be maintained. Then you see there is an inlet for sterile air. This air is passed on into the fermenter through a structure called sparger. This sparger is actually a plate that contains holes in it. What happens is that when you pass air, the sterile air from through the sparger, it gets pressurized, and that pressure also is an additional mixing device which will mix the contents of the fermenter. Plus, it also creates bubbles in the contents of the fermenter, which increases the surface area for microorganism to have more oxygen for respiration. From down here, you can collect your product or your harvest. Sterile steam is just entering before you collect your product. It is maintaining the aseptic conditions. Then we have a cooling jacket. From here, the cool water is entered, and from here it is taken out. The purpose of fitting a cooling jacket here is that to control the temperature. This may contain water, cooled water, or the pipes of cooled air. The reason or the purpose of putting them here is the same, that is to control the temperature. Now we have the inlet for microorganism and nutrients. They will be different for different products. For example, for penicillin, and penicillium is used, which is a fungus. Nutrients will accordingly be changed according to or depending upon whatever product you are forming. This nutrient and microorganism will be placed into the fermenter through this inlet. Okay, this is the description of all the structural features of the industrial fermenter that I showed you in this diagram. All these are described here in this slide. Nutrient inlet will allow the sterile nutrients to be added. Nutrients are obviously required for the growth and reproduction of the microorganism that we are using. Mind it that the nutrients must be sterile so that your product form is not contaminated. Aeration system is there, which is allowing air to pass through under pressure through the sparger so that bubbles increase the surface area, which facilitates an increase in respiration rate. This oxygen is obviously required for the aerobic respiration of the microorganism. Then we have sometimes filter adjusted here, which maintains the aseptic conditions, preventing the entry of microorganism. Cooling system is there to maintain the temperature since microorganisms grow best at an optimum temperature. Plus also the product formed are sometimes temperature sensitive. Increasing the temperature might cause problem in your product. pH control is again required for microorganism maximum growth. pH and temperature probes will detect the changes in pH and temperature in the contents of the fermenter respectively. Steam inlet again maintains the aseptic conditions. Impeller or the stirring device will agitate the fermenter contents, mixing the nutrients and oxygen well within the fermenter and keeps the temperature even. Now we have to learn about two basic operating systems which are used in industry in the fermenter. Either the product is formed from batch operating system or it can be formed from continuous operating system. What happens in batch operating system is that you add your nutrients, you add your microorganism into the fermenter, you start the process for a specified period of time, and after that you take out all the nutrients and microorganism from the fermenter, whatever left is. You take out your product and then you sterilize the fermenter, clean it well, and then start the process again. On the other hand, in continuous operating system, the fermenter is never emptied or sterilized. Instead, the nutrients and microorganisms are continuously topped up. They are being added from the above in the fermenter, and your harvest or your product is continuously removed. The sterilization process doesn't happen here. Both the processes have their advantages and disadvantages. Batch operating system is easy to set up and easy to run. The vessel can be used for different processes at different times. In case of contamination, it is easier that you detect the error in one batch, and only that product would be discarded which is causing you the problem. The other batch will go unaffected. On the other hand, in continuous operating systems, smaller vessels are needed, greater productivity is there, and it is more cost effective. Now we have to see the disadvantage of batch and continuous operating systems. The disadvantage for batch operating system is that time and cost is required for sterilization. The fermenter needs to be sterilized after every batch. And for this operating system, you need to have a larger vessel. On the other hand, continuous operating systems are more expensive to set up and more difficult to run. Practical problems includes clumping of the cells of the microorganisms. The growth of microorganisms such as that of fungus may block the inlets or may start to form foaming structures which may cause problem. In case of contamination in continuous operating system, it gets very hard to detect at what exactly the error occurred, and the entire culture is to be wasted if the error comes or the contamination comes.